Bismillahirrahmanirrahim. Assalamu alaikum dear students. Today we are going to discuss the protein synthesis. As we know that the DNA which contain the genes and they have to express themselves. When they express themselves, they form the protein. So the whole process take place inside the cytoplasm. So here we have the double-stranded DNA which make their copy, and we can simply say that they form a single-stranded RNA, and from the single-stranded RNA a protein is formed, right? So the process in which the double-stranded DNA which form the single-stranded RNA, this is known as transcription, while this messenger RNA then form the polypeptide chain of protein, right? So this process is known as translation. So the whole process in which the transcription and translation take place, this is known as central dogma. So that was given by the scientist Francis and Crick, this process is basically the copying information from the one strand of DNA into the messenger RNA, and then from the messenger RNA the protein synthesis is formed, right? So it is the process in which the enzyme which involved are known as RNA polymerase, right? So transcription basically takes place in the nucleus of eukaryotic cell, and the strand of DNA which contain the polarity of like 3' to 5', that direct the synthesis of RNA. That strand which play important role in the copying which is going to be copied, this is known as template strand or antisense strand, and from this antisense strand the next strand which is formed, this is known as sense strand, right? So other strand of the DNA with the polarity would be 5' to 3', which is sense strand, right? This is also known as complementary strand or template strand, right? And we call it the coding strand or sense strand, right? These are the three basic names. So that is very important. So the genetic code present in this strand is similar to the genetic code which is present on the RNA basically, except the uracil, because the DNA contain the 4 nitrogenous bases: adenine, guanine, cytosine and thymine. But when the DNA is transcribed into the messenger RNA, or when the messenger RNA is formed, so this uracil, thymine is replaced by the uracil, right? So it means that the coding strand of DNA has the same sequence of nitrogenous base as the messenger RNA. So here it has the same polarity as the RNA transcript. Now the transcription unit, so it is defined as the segment of the DNA between the site of initiation and termination of transcription by RNA polymerase. So the promoter is the region on DNA molecule, right, to which an polymerase enzyme have to bind. So promoter is located upstream to the left of the structural gene and towards the 5' end of the coding strand, 3' end of the template strand, right? Now location of the promoter gene and terminator gene is always mentioned with reference to coding strand. Now look at the diagram, you can easily locate the promoter, right? So it has the different part for attachment to various transcription factors. So in most eukaryotes the promoter has an ATP rich region which is known as TATA box, TATA, thymine adenine box. Now next, the area has groove on to which the specific protein components can combine. Now this Tata box is known as Pribnow box in prokaryotes and Hogness box in eukaryote. This point is very important, right? So beside the promoter, eukaryote also require an enhancer which enhance the activity, which increase the activity, right? So the second part is the structural gene, the area of the template strand that is involved in the transcription or formation of the RNA are known as structural gene, right? So they are of two types, monocystronic and polycystronic. So mono mean one, cystronic, these are the sequences which help in the formation of polypeptide. So it is made up of only one cistron, while the polycystronic are made up of many cistron. Now monocystronic carries information for synthesis of one polypeptide chain, while the polycystronic they contain the information for synthesis of more than one polypeptide chain. Poly mean many, right? So the third point is that monocystronic is mostly found in the eukaryotes, while in prokaryotes the polycystronic region are found, right? So the third region is the terminator. So this is the region on DNA molecule present downstream of the structural gene, that is toward the 3' end of the coding strand, 5' end of the template strand. So terminator region defines the end of the process of transcription. So the next is the transcription unit and the gene. As we know that the gene is a functional unit of the inheritance, and gene are located on the DNA, and the DNA sequence coding for the transfer RNA or ribosomal RNA molecule are also defined as a gene. So cistron is defined as the functional unit of gene. It is the segment of DNA coding for a polypeptide. So the structural gene is a transcription unit basically, and it is monocystronic in eukaryotes and polycystronic in prokaryotes. So the monocystronic structural gene have interrupted coding sequence. The gene in eukaryotes are in split manner, right? The coding sequence or express sequence are defined as exon, which appear in mature or processed RNA. It means the exons are coding sequences of the DNA. Now the exons are interrupted by the intron, mean there is a exon intron, exon intron, exon intron. So between the two exons there is a sequence which is known as non-coding sequence of the DNA, which is known as intron. So the introns are intervening sequences that do not appear in the mature or processed RNA. So simply here we have a process which is known as RNA splicing, right, in which the introns are removed, right? So again the exons are joined together after removing the introns. So this is known as RNA splicing. So the split gene, right, arrangement further complicates the definition of a gene in terms of a DNA segment. So we simply are very much confused in this term. Now the next is RNA synthesis. So the mechanism of RNA synthesis was worked out in the late 1950s by Howitz, Weiss and Steven by in vitro experiment. No primer is needed for the RNA synthesis basically, right? So primer are the sequences, are basically the nucleotide sequence that initiate the process. So in RNA synthesis there is no need of primer. So material required for the transcription are enzyme, RNA polymerase, DNA template, 4 nucleotide and divalent metal which act as a cofactor. So the RNA polymerases are of basically different types, 3 basic types, right? In prokaryotes there is only single RNA polymerase is used, while in eukaryote there are three RNA polymerase which are synthesized, right, that are required for the process of RNA synthesis. The RNA polymerase 1, 2 and 3. In eukaryotes these 3 are involved in the synthesis of RNA, while in prokaryotes there is only one single RNA polymerase is required. Now RNA polymerase 1 synthesizes the ribosomal RNA in the nucleolus, RNA 2 synthesizes messenger and hRNA, and 3 synthesizes the transfer RNA in the nucleolus. Now RNA polymerase is basically a very complex structure. So here is the structure of the RNA polymerase. It consists of the core enzyme which has 4 subunits, and which include the alpha, 1 beta and 1 beta prime, right? So the 1 alpha, this bind to regulatory prime protein basically, and the beta subunit bind to the DNA template, and the beta prime subunit bind to the RNA nucleotides, right? So the second part is the sigma subunit which recognize the promoter and initiate the synthesis basically. The third is the Rahu segment, help in determination of transcription. Now the process of transcription in prokaryotes, it includes the 3 basic steps: the initiation step, elongation step and termination step. This process takes place in the cytoplasm, and it is the DNA dependent RNA polymerase enzyme which play important role in the process of transcription in prokaryotes. All three types of RNA are needed to synthesize a protein in a cell. In prokaryotes, the structural genes are polycystonic and continuous, right? So these were the requirements which were needed for the transcription in prokaryotes. Now we will study the three steps separately and in very detail. The first step is the initiation step. In first step, the RNA polymerase attached to the DNA with the help of the sigma factor. So this sigma factor recognizes the promoter region, right? So then RNA polymerases initiate the transcription after the recognition of the promoter region. So it uses the nucleoside triphosphate as the substrate and polymerase in a template dependent fashion following the rule of the complementarity, right? So it also facilitate the opening of helix. When it binds the DNA, it open the DNA strand. In elongation step, RNA polymerase in the presence of energy and magnesium ions can catalyze the process of elongation, right? So here the magnesium act as a cofactor. As the enzymes move along the DNA template, RNA chain become longer. Synthesis of the RNA continues till the enzyme reaches the terminator, right? Next step is the termination. When the RNA polymerase reaches the terminator region, a specific chain terminating protein called Rahu factor, also called termination factor, stop the synthesis of RNA chain. It separates the RNA polymerase as well as the newly formed RNA strand. RNA and RNA polymerase fall off and it results in the termination of transcription. So important points are the messenger RNA does not require any processing to become active. Transcription and translation take place in the same compartment as there is no separation of cystole and nucleus. Many times the translation can begin much before the messenger RNA is fully transcribed. So these were the important points, because as we know that in prokaryotes there is no nucleus, so there is no distinction between the nucleus and the cytoplasm, so that's why the whole process takes place in the cytosol, right? So these were important points. Now the next step is transcription in eukaryotes. How transcription takes place in the eukaryotes? It is more complex than that of the prokaryotic transcription. So it takes place inside the nucleus and of two types of semi-autonomous cell organelles, mitochondria and plastid, because there is a DNA which is also present in the mitochondria and which is known as mitochondrial DNA, and there is also a DNA present in the plastid, right? So it occurs in G1 and G2 phase of the cell cycle. So special features of the transcription in eukaryotes are, so the transcriptional unit has only one gene, mean it is monocystonic. The second step is that initiation require protein called transcription factors. Sigma factors is absent, right? No primer is required. RNA are released and processed in the nucleus, and coupled translation is not possible because in eukaryote the nucleus and cytoplasm are present separately, right? So the greater part of the product pass from the nucleus into the cytoplasm, mean transcription take place in the nucleus and messenger RNA comes into the cytoplasm. Now the processing of the primary transcript, which is messenger RNA, the nascent or newly formed messenger RNA by the RNA polymerase 2 is called hnRNA or primary transcript. The primary transcript contain unwanted base pair or sequences which is known as intron, and useful base sequences are known as exon. So the primary transcript is converted into functional messenger RNA after post-transcriptional modification. Following steps are involved. Number 1 is capping, number 2 is tailing, number 3 is splicing, and number 4 is ligation. Now, capping of the 5' end, it is addition of cap to 5' end of the messenger RNA, right? So it occurs shortly after the start of the transcription. The cap contains unusual nucleotides, methylguanosine triphosphate. Methylated caps protect the messenger RNA from degeneration by nuclease enzyme. So it also provides a ribosome binding site. Tailing at 3' end, this is known as polyadenylation. It is addition of a poly A tail. Poly A tail means adenine tail at 3' end of the newly synthesized messenger RNA. So this poly A tail contains adenylate residue. This is about 200 to 300 adenine group is attached over there. Now the next step is splicing. So the splicing is the process of removal of introns through cutting and joining of exon. Now the next step is ligation. So the end of the adjacent exons are now joined together by ligase enzyme. The S and R and Ps are formed by the association of protein with small nuclear RNAs. Fully processed H and RNA, which is newly RNA, messenger RNAs, is released from the nucleus into the cytoplasm for translation. Now the next is split gene arrangement. It represents the probably an ancient feature of genome. The presence of the intron is reminiscent of the antiquity, and the process of splicing represent the dominance of RNA world. Now the next is in vitro synthesis of RNA, how RNAs take place in vitro. So Korenberg for the first time introduced this RNA in vitro. Now the next step is the reverse transcription. How reverse transcription take place? This process is also known as Taminism, so because it was first discovered by Tamin and Beltmore. The process of formation of DNA from RNA is known as reverse transcription, right? So a special enzyme called reverse transcriptase help in this process. RNA is the genetic material in some viruses like TMV, tobacco mosaic virus, bacteriophage, HIV, etc. So the viruses that have RNA as a genetic material are known as retroviruses. These inject their RNA into the host where this RNA synthesizes a DNA copy. It gets incorporated into the host genomic material. This viral DNA is transcribed into messenger RNA and then translation takes place again. So RNA form the DNA through reverse transcription, and again there is a transcription of that DNA take place and formation of messenger RNA, and then again protein take place. So it is the newly synthesized viral DNA gets active immediately. Then the virus multiplies in the host and the host cell ruptures. This is known as lytic cycle. Sometimes the viral DNA gets incorporated in host DNA and remains inactive for some time. It replicates with the host DNA. This is known as lysogenic cycle. Hope you liked my video. Kindly subscribe my channel. Thank you.